Oh, wow. Like that. Look at that. Michael, impressing from the start. So Michael is a PhD student in the computational track of the molecular genetics department at the University of Toronto, and also I've never known how to pronounce this, but Lunenfeld. Maybe you can correct me in a moment.
Lunenfeld.
Lunenfeld, Tanenbaum Research Institute, and he works with Kieran Campbell. And so he's very interested in pancreatic cancer, and he's going to talk about Snakemake. And so thank you very much, Michael.
So thanks for the kind introduction, and also thanks for the opportunity to present here today. Essentially, I work in the biomedical field, and I'm going to be talking about how I build pipelines and also reuse them, in particular, focusing on how to do this when data changes and parameters change between data sets. And so just a brief overview, my own work focuses on the cancer immune system relationship. And so this relationship is sort of summed up in this image quite nicely. So in the center, you see a cancer cell, and on the outside, you see what are known as cytotoxic killer T cells. And so these cells literally go ahead and kill cancer cells. And this relationship is quite important, in part also because it, one, protects us from cancer, but two, therapeutic agents target this relationship now. And these are relatively new, but also have shown great success in some cancer types. And so some of the specific questions that we're, for example, interested in is how many immune cells are present in a cancer biopsy, or what states are these cells, the cancer cells and immune cells in, and what variability exists in the cancer cell population in general, but also what happens to them as we perturb them. And it's only been fairly recently that we can actually answer these questions. And a lot of that is driven by a reduction in the cost of sequencing. And so I assume that many will be familiar with Moore's law, which essentially states that the number of transistors on an integrated circuit doubles every other year. And on this graph, you have time on the x-axis and cost on the y-axis, and Moore's law is overlaid over here, shown as a straight line on a log graph. And then in green, you have the cost of sequencing a full human genome. So back in the early 2000s, when the first human genome was sequenced, this cost the funders about $100 million to do. And the cost has been dropping at a rate faster than Moore's law ever since. This figure is a little outdated. By 2013, the price was about $5,000 per genome. And at this point, it's probably below $1,000. So perhaps unsurprisingly, with this reduction in the cost of sequencing came an explosion in the amount of data that is publicly deposited. So here you have the terabytes of data deposited in the short read archive database, which is essentially one of the databases where lots of sequencing gets stored. And as you can see, in 2019, there's almost been 400,000 terabytes of data deposited of sequencing data. And perhaps also unsurprisingly, data sets of this magnitude will require robust pipelines to process it all and make sense of what is essentially all A, C, T's, and G's. And before I get into how I do this now, I want to take a look back and reflect on how I used to do this. And this is also a tale on how not to do it. So about 2 years ago, I was still an undergrad at a different institute. And so one of the things we had was this nice robot over here. And ironically, and arguably, this is probably the most reproducible part of my workflow. So essentially, there's a bunch of pipettes here, and then these are what are called 96-well plates with a sample in each well. And so the robot then automatically handles this, and this cuts down on both cost and time it takes to process these. And so we did quite a lot of this, and then we ended up with about 1,000 samples from diverse cancer models using this robot, and it was partially my job to make sense of all this data and process it and clean it. And so what I used to do is to submit data cleaning jobs manually. This looked something like this, so I started writing out things in Bash. This was fairly big data, so it all happened in a computing cluster. And I essentially looped through all my data that was in a directory, wrote a batch script, and submitted this to the cluster with different parameters. And of course, 5 months down the line, I had trouble figuring out what parameters I used for what file, even though I sort of semi-tried encoding this in the actual file name, but it wasn't great, this the summary. I also rewrote a lot of code. So every single time the robot ran and we got new data, I would go back to the drawing board and essentially type out all these commands again. There was also a lot of R scripts that I had and functions that accomplish individual tasks, but they were not linked to any one project. And so I would individually run these whenever I needed any one of them. And again, 5 months later, it was kind of hard to figure out what I did when. There's a few other issues with this approach too. One of them is that changes in the upstream steps. So for example, if I decided that I wanted to change my data processing somehow, those didn't trigger a rerun of all the subsequent steps. So if I forgot to change one of the steps in between, then I would essentially have a mistake in my code somewhere. And overall, the code usability across all of this was quite poor. So if you think a little bit about what we would like a pipeline to be like, first thing is probably reproducible, and we can talk a lot about this. In fact, we have a whole 3-day conference about it. But the things in practice that this means for me is to set a seed whenever randomness is involved, and also to add relative file paths. We also talk a lot about whenever we write code, the idea is that if you write your code more than twice, you should probably write a function for it. I like to think that this should be extended to pipelines. Whenever you use the same script more than once, you should probably package it up into a pipeline and not have the same script doing slightly different things all over and over just because you changed a parameter somewhere. This script should also be tied into a single executable workflow and ideally create a results report at the end that can then be shared quite easily with a collaborator. They should also rerun workflow sections automatically if there's upstream changes. So for example, if I have this toy workflow here of downloading data, clustering some of it, and then creating a figure, if I decide that I want to change my clustering parameters and I rerun this step, the creating the figure part should automatically rerun without me having to do much or even think about it much. And then finally, I also would like to be able to reuse the same pipeline on different data sets. And so the example I'll showcase in just a bit is essentially quite a complex data processing pipeline. And the problem here is that most, if not all of the code remains the same. And it's only the parameters that change from dataset to dataset. And importantly, sometimes I can't even programmatically define these parameters. I need to go look at plots to figure out what these parameters should be, and only then can the pipeline proceed. So the example where I've run into this issue was of processing single-cell RNA sequencing experiments. And so here, the data modality doesn't matter too much, but essentially it is RNA sequencing. What the RNA from each individual cell is sequenced, and this allows us to understand the heterogeneity that exists between cells. And these are, again, some of the questions that I listed earlier. And this is also the kind of data that allows us to answer these questions. So to outline some of the steps in this workflow, the first step is to really filter out any dead and damaged cells. So the wet lab technician or whoever is actually running the experiment, throughout that whole process, cells will die, some things will not work. And these are cells that we don't want to keep in our analysis pipeline. And so there's really two ways to filter them out. One of them is to filter out cells that have very few reads in them. And so a read, the technical definition doesn't really matter here, but it's essentially how many times all of the different RNA molecules were counted in that particular cell. And then there's also reads that come from mitochondrial genes. And we want to keep that threshold somewhere. But these thresholds are data dependent. So for example, if I have a sample from cancer cells, then this mitochondrial threshold should be higher than if it were normal samples. And so this is an example plot from one of the data sets that I have processed. So each point here is essentially a cell. And then on the x-axis, you have the number of reads that were detected in that cell. And on the y-axis, you have the percent of mitochondrial genes that were detected in that cell. And so for this particular example, I set a threshold of 1,000 detected reads. So anything above that I kept, and everything with less than 20% mitochondrial gene counts I kept too. So essentially this rectangle here, those are all the cells that I decided to keep. But importantly, I wouldn't have known about this before looking at this plot. So I can't create an automated pipeline to run all of this because there's no way for an algorithm to figure out where to place these lines, and it's more of an art than a science sometimes. So that's the first data-dependent parameters that I need to infer. The next step is generally to normalize the data. This can be as simple as log normalizing them. Then cells are clustered, and then I assign each cluster to a cell type based on the expression of certain marker genes. So different types of cells have markers that are expressed only in those cells. So for example, cancer cells have one set of markers, and immune cells have other set of markers. And based on these markers, I then assign each cluster to a cell type. But of course, the problem is that some clusters are impure and contain multiple cell types, in which case I would want to take just that subcluster and recluster it. Again, this is something I don't know before I start implementing things, and I really need to wait and see and look at plots to determine this. Something else that is quite helpful is to look at scatter plots between different markers. And then based on this, I can assign cell types to each of those clusters more easily. But again, which two markers to correlate and to look at depends on all of the previous plots. So just a summary of some of the data-dependent parameters. And this is not all of them, but it is the main ones, and it sort of gets the point across. The first one is the number of detected reads and the number of mitochondrial genes, the clusters that need reclustering in the IDs, as well as the markers to correlate with each other. So in summary, we really want a pipeline that is reproducible, that if I run it again, I get the same results, that I can execute in a single workflow, and that reruns the same script over different samples or parameters if that is necessary. So for example, if I want to plot the expression of a gene across all the different clusters, I don't want to do that. I don't want to recreate that script many times. I want to have one script to just plot these things and then rerun that same script. I also wanted to automatically rerun if there's upstream changes, and I want to use the same pipeline despite these data-dependent parameter differences between datasets. And so the way I implement all of this is using Snakemake, which is a Python-based workflow manager. And it's essentially based on rules, which are human readable code snippets. These can be tied together to run locally on a cluster on cloud environments, and should be easily transportable between any of them without really needing to modify the workflow. And so a basic rule essentially looks like this. It starts out with the word rule, and then it has a name. This name needs to be unique, but other than that, there's not really any requirements. And then it has an input and an output. So this is where the user can list what data goes into the rule, what it should spit out, and then how to do it. And in this case, that's just an R script, but it could just as well be a Python script or a shell command or something. And then I can tie lots of these snippets together to create a complete workflow. And this looks something like that. So I'll guide you through this essentially from the bottom up. The first rule might be to download a bunch of data. So there's also a parameter keyword where I can specify where I would like to download this from. I specify the output file name, and then a simple shell command can download this from this website and save it as this output. The next rule essentially takes this output as its input, and then it creates a figure from this using my R script. And so if I were to just have these two rules on their own, Snakemake wouldn't really know what to do with it. The other thing that's required is a special rule called rule all. And so here I list all of the things that I want Snakemake to create. In this case, it is a process data CSV and a figure. And then Snakemake takes a look at this, and then it tries to find all of the rules that create this. And based on this information, it creates a directed acyclic graph where it starts at rule O, it checks, oh, I need this figure to be created. That is created by this plot figure rule, but this plot figure rule needs the process data as input. So actually what needs to run first is download my data. And then when I run the whole pipeline, it starts by downloading the data, plotting it, and then in this case, it's done. But this can be expanded to an arbitrary length. The other thing that is quite nice is that I can rerun the same thing quite easily. So let's say, for example, I want to plot the same figure, not once, but 3 times, once for each sample. One way to do this would be to list out the sample every time, or I could also use expand command in Snakemake that essentially takes a path with a wildcard and a file name, and then also another list that contains values, which in this case specify the samples. So this would return a list with 3 file paths and names with my amazing figure -S1, S2, S3, etc. And then the only thing I need to do to the other workflow I showed earlier is to include these wildcards everywhere where there's data. And then Snakemake automatically knows, oh, I need to download 3 samples from this website, and I need to create 3 figures, and it just runs it. The other thing I would briefly like to talk about is a configuration file that can be added quite easily in essentially just this fashion. And this is just a YAML file with configuration. This is generally a good place to keep file paths to data or software binaries. This is also where I keep all of my data filtering parameters. And the other thing is conditional rule execution. So, for example, the sort of classic example of this is to, for example, plot the expression of a gene in all the clusters. If I don't know how many clusters there will be, it is hard for me to specify what clusters to plot this gene in. And so Snakemake solves this using checkpoints, essentially, where it creates a directed acyclic graph all the way up until this checkpoint. It runs all of that, and then it recreates the iterative acyclic graph once it knows the output from the clustering script. But this still leaves one problem, which is this only works when Snakemake can programmatically determine the next results. What happens if I need to determine these next steps based on manual inspection of plots? And I sort of mentioned some of these examples earlier. But these are situations when thresholds, for example, are data dependent and require manual inspection. And the first example I've showed was this plot where I needed to find thresholds for the number of detected reads and the percent of mitochondrial genes in a cell. And so the way around this that I found is to essentially start my snake file with some configuration where I save all of these parameters in, and then to create a dictionary that at the beginning contains all of the outputs that Snakemake can generate automatically. In my configuration file, I will then also have parameters for these thresholds. And so when I run this for the first time, they're empty. So all of this evaluates to none. So none of this will run. So we can ignore it for now. And then I have my rule O, and here I put as input, I have all of the values from this dictionary. And so in this case, Snakemake can just run ahead. It generates everything it can generate automatically. And then at the end, it automatically sends me an email and says, hey, everything is done. Please come back and check your plots and determine these thresholds. So then I log back in, look at my report, and look at these plots and say, okay, this is where I want my thresholds to be. I go into my configuration file, put those in, and then I rerun Snakemake. And now, because this configuration parameter is not none anymore, this evaluates, and I add things to my dictionary. And once Snakemake gets to running again, it notices, oh, the results are downloaded. This doesn't need to be rerun, but there's this process data file that isn't there yet that I need to recreate. And so essentially then it runs that rule to create that. And the rules can exist without issue. Okay. The final thing I wanted to mention is the report. So this is quite a neat feature where essentially all one needs to do is type snakemake -report with a name. And then this creates a report of this kind. So what you see here is essentially the directed acyclic graph again, starting at the very top, and each of these names is a role, the name of which doesn't really matter for the sake of this presentation, except to say that this is actually an actual example of one of my reports that I've used to process one of these data sets. And then on the left-hand side, You also see statistics and configuration, as well as all of the results. And so one can mark plots to be included in this report within a Snake file, and then they would be included right here. So to take a look at some of these, the statistics tab essentially shows the runtime for each of these rows in seconds. And so each point is one of the times this ran. So plot marker density maps, for example, runs once for every marker, and I have about 100 markers in my configuration. And so this run 100 times, which is why there's so many points. And then I can also see a creation date of when this rule was last run and from what date that output is from. And then my configuration tab essentially just copy paste that YAML file that I used as a configuration. And so if I ever am confused about what number I used as any one of these thresholds, I can just go back in here and check what was used. Now to the results, this is essentially what those look like. There's a thumbnail of whatever figure I decided to include. I can just click on it, it enlarges, I can download it from here. And then there's also the parameters and wildcards that were used as part of this. So to summarize, I use Snakemake's pipelines to create reproducible workflows. It's quite easy to just put these on GitHub and then clone them elsewhere. It just runs whether locally or on the compute cluster. I tie all of these rules together to create a single executable workflow. And then I can also rerun the same script over many different parameters and all samples using the expand function. I can also automatically rerun things if there's upstream changes. So Snakemake automatically detects these based on timestamp. And then finally, I can reuse the same pipeline despite data-dependent parameter differences between data sets, either through checkpoints, if these can be evaluated programmatically, or using if-else statements and then adding them to a rule when these parameters must be specified manually. Snakemake is quite powerful, so there's many more features that I didn't talk about, some of which are Docker containers. So each rule could have a separate Docker container. There's also a possibility to add one Docker container for the entire workflow. It allows for the specification of Conda environments. There's also wrappers for very commonly used tools that one can just use online, and then it doesn't take a lot of installation. There's support for log outputs and also high performance computing specifications such as the number of threads or the memory required. For anyone interested, I would highly recommend both the tutorial and the documentation. There's always new things that get added. And yeah, my slides are here in case anyone wants to look at them afterwards too.
Thanks very much, Michael. That was amazing. Does anyone have any questions for Michael? I'll just get us started with a quick one. How much time and heartache do you think that this has saved you since implementing this system? And if other folks have questions, add it to the chat or raise your hand. And
yeah, no, I think quite a lot, and it's only the beginning, I think. So this started with a collaborator asking me to process one of these data sets, and I did that, and then they came back and were like, okay, great, can you do this with another data set? And then I had to do it for my own data set. So I think in total, I've processed about 3 data sets with this now. And now that it's up and running, it works quite fast. And yeah, I think probably a few days at least, but now this has also reduced the barrier of entry to just getting more data sets, right? And so it definitely speeds things up.
Periscope?
Hi, Michael. Thank you for the presentation. Very interesting. I have 2 questions. The first question is about the part of the work that needs some manual inspection, as for example, checking the visualizations and establishing the thresholds. I think you mentioned that, but I might have lost it. How do you actually incorporate this manual work in your system?
Yeah, so that's the thing, right? I can't incorporate this automatically because it is just a very manual process that I don't know how to automate, and I don't even know if it's possible to automate. So in that case, what I used to do before is to have an automated part, and then I used to sort of add to it manually, and then I copy paste things from elsewhere, and then hard code all of the parameters once I saw the plots, right? But the whole idea of this is that there's a parameter section where I can add these parameters once I've determined them, but the pipeline only ever runs in increments, and it stops at the place where I need to try and find these, right? And then I manually inspect the plots. I add those thresholds to the configuration, and only then is it allowed to keep running.
Thank you. And the other question was about the Docker. I'm very, very new. Do you have any recommendations, like from your experience, about the Docker?
Yeah, it really depends on what you want to do and what you want to run. So I just create my own Docker containers. I usually start out with the bioconductor base Docker container, and then I add stuff onto that. But yeah, it's kind of case dependent on what you want to do.
Okay, thank you very much.
Yeah.
Devin in the chat is asking, what are the advantages over a bash script file?
Yeah. Well, this is how I used to do things, right? I had my little bash scripts. There's many. One of which is sort of just essentially, if you have several bash scripts that all run one after the other, either you tie them all together, or what I used to do is to run them individually, but then you need to remember to run them individually every single time. So Snakemake essentially handles all of this for you and runs it automatically.
Are there any other questions or comments? Right, well, just say thanks once again, Michael. That was very interesting.
Thanks.